Sometimes we take our lives and our health for granted. We get upset at little things and worry about the inconsequential. We create all these plans about our future and what we want to do without knowing if there's a future in store for us. Imagine going into the doctor because you have a headache and have been throwing up. Now imagine leaving with the news that you have a malignant brain tumor. All those fights seem stupid. All those words mean nothing. And all those plans seem impossible.
Journey to unknown. Look at us now, how we've grown.
Like you see up on the left, you can see that there's an abnormality there, and those of us who've looked at a lot of these brain tumor scans understand that that's a brain tumor there. The short arrows are pointing to the area where there's actually a brain tumor, but you can't really tell where that begins and ends. And if you look at the long arrows, those are pointing to the primary motor strip. The primary motor strip is what drives every movement of our hands and legs and our breathing muscles.
You want to take out all the tumor so the tumor doesn't recur. If you leave behind pieces, then they will regrow and form a new tumor. But you really can't take much more than the normal tumor, so because you're probably going to cause permanent damage to the patient whose brain you're operating on.
Unfortunately, Violet's succumbed to her tumor. She had a type of tumor called brainstem glioma. And in the 22 years that I've been taking care of kids with cancer, I've never had a child with this kind of cancer survive. And as you can imagine, those are the most difficult first day conversations that you could ever have with a family. And she and her family met this news with aplomb. This is after she was diagnosed. And you can see her sassy nature came through in every single conversation we ever had. And her family's wonderful spirit came through at every interaction we ever had. And Violet knew about our Avatar mouse program, and she knew that we were using that to study pediatric brain cancers. And she said, you know, nobody with my kind of cancer ever survives, and nobody with my kind of cancer ever gets a surgery because it's in the brainstem, and so you can't do surgery. So how are you going to ever learn about my kind of cancer with other kids? And I said, right now we can't. I'll be honest with you, we don't have any mouse models, we don't have any cell lines. And she said, when I die, I want you to do an autopsy, I want you to take my tumor and I want you to make a mouse, and I want you to help other kids with that.
But there may be a solution. The Olson Lab at Seattle's Fred Hutchinson Cancer Research Center has come up with an innovative way of aiding surgeons operating on tumors. They have created a molecular paint that coats cancer cells. We headed over to Fred Hutch to learn more. In their first experiment, the lab took a human glioma and grew it on a genetically engineered mouse. They took a peptide from the Israeli deathstalker scorpion and attached it to a molecular flashlight. Then they injected this into the mouse's tail vein.
It was the craziest thing. We injected the mouse, and about an hour and a half later, the tumor started glowing like crazy, and the rest of the mouse wasn't growing. If you want to see grown men dancing and jumping, that was the day. That was the eureka moment. Right?
Chlorotoxin, it's one of many dozens or hundreds of proteins that are in scorpions. Its role in venom is mostly isolated to small animals like shrimp. It's not believed to be toxic at all in mammals like us or mice. The chlorotoxin molecule is only about 37 amino acids long, so it's very small. It's unusually small for many proteins that are expressed in the body. Its very small size gives it a couple benefits. One, it folds into a small, very stable knot, allowing it to exist in the bloodstream for a fairly long amount of time, whereas many other proteins get chewed up in the blood. And because it's so small, it works more like a drug in the way that it moves through the body and gets to tissues than it would like a much larger protein. So that made it a great candidate as a basis for a therapy. Then the fluorescent molecule, in the first generation of Tumor Paint, we've used Psi 5.5, which is this near-infrared fluorescence. We chose near-infrared because it has a low background in the human body and it has an ability to get through small amounts of tissue. So if there were, for instance, a small piece of tumor behind a fold of normal brain, the surgeon would have a chance to see that.
These are two brain tumor cells, and you can see inside the brain tumor cells hundreds of copies of tumor paint, even 15 minutes after we expose these cells to the tumor paint. And these are normal skin cells, and you can see that there's no copies of tumor paint in those.
Here's a study on tumor paint that was conducted in the Olsen lab. There are 3 mice, 2 with tumors and 1 without. Out of the 2 mice with tumors, the vehicle mouse has no treatment, while the other does. The mice in the image get tumors because they have overactive growth signaling pathways called sonic hedgehog, telling the tumor cells to grow. IPI-926 is a drug that inhibits sonic hedgehog. Therefore, IPI-926 can be used to treat the tumors. The different colors of the tumor paint show how intense the tumor paint signal is. Yellow-red is bright, purple-black is dim. The tumor paint helps light up areas that otherwise would have been difficult to see. Notice that the drug did not completely eliminate the tumor. This tumor still would have come back even if it was continuously treated with the drug. The last row is H&E staining. It's a classic way that doctors will look at slices of tissue. The purple is cell nucleus and the pink is the rest of the cell. Tumor cells are mostly nucleus, so you see a lot more purple where the tumor is. Some cells in the cerebellum in the wild-type normal mouse are also mostly nucleus, and you can see that on the bottom right picture, but this is normal. After looking at these various images, it's clear that the tumor paint gives the most detailed picture of where the tumor exactly is.
One of the really exciting things that we found is this is not just something that lights up brain cancer. It lights up a wide range of cancers. Prostate cancer, colon cancer, skin cancer. So it may be something that could be widely used in many different types of therapy for many different types of cancer patients.